Hello everyone, thank you for being here. The title of my talk is Integrative Computational Approaches for Multi-Target Drug Discovery in the Arachidonic Acid Pathway. Here, I will talk about our work that we did together with our Dean, Professor Dr. Erdem Balaban. So I will start my talk by discussing Arachidonic Acid Pathway, its role in various inflammatory diseases, and some of the key targets that we are focusing our studies to suppress production of various arachidonic acid metabolites. Later, I will briefly discuss our hit-to-lead journey and talk about our visual screening studies that conducted, and I will share our focus on designing better visual screening workflows to screen chemically distinct chemical compound containing libraries with different sizes, which can result in different outcomes. By the experience we gain from chemical derivatization studies performed in our organic chemistry laboratory, the observed structure activity data from internal and external data sets let us develop more advanced hypotheses to be tested in further studies. Together with the application of physics-based and AI-driven approaches, we reached some important preliminary findings, and I will be happy to share the results with you. So let's start to dig into the details of arachidonic acid pathway a little bit. This is a key pathway which becomes activated in the event of an inflammatory stimulus. Cytosolic phospholipase A2 acts on membrane phospholipids, hydrolyzes them to arachidonic acid, and some other lysophospholipids. Arachidonic acid is further metabolized to eicosanoids. You can see the metabolism of our prostaglandins on the path, which is located at the left-hand side. In the middle path, you see the metabolism of leukotrienes, and on the right side, we see the metabolites called epoxy-eicosapentaenoic acids, which are generated via cytochrome enzymes and further metabolized to inactive diols by soluble epoxide hydrolases. So here, you might have already recognized that cyclooxygenase 1 and 2. So, as you very well know, non-steroidal anti-inflammatory drugs that target those enzymes are one of the most prescribed drugs that are in the pharmaceutical market, and they are used for treating different inflammatory disease conditions. But it's hard to use them in chronic inflammatory diseases because of their side effects. Therefore, there is a need to find new regulators that target the other biological targets on the path, let's say mostly inhibitors, which might result as hopefully better inflammatory treatment solutions. So on the prostaglandin pathway, the path starts with the cyclooxygenases, as you can see. The prostaglandin metabolites of the PGE synthesis, which produces PGE2, and that's related to inflammatory response. If we can block the pathway downstream instead of blocking it from the top, we might achieve more specific inhibitory treatment options, since we will potentially reduce the side effects that are related to thromboxanes and also other metabolites of arachidonic acid. Another option of blocking the path could be via lipoxygenases. By inhibiting 5-lipoxygenase or its activating protein, which is called 5-lipoxygenase activating protein, shortly FLAP, inhibiting the metabolic path from here might be another option not to deal with the side effects of COX inhibition. And that could be an alternative approach. An additional approach might be the inhibition of soluble epoxide hydrolase enzyme. This enzyme metabolizes the fatty acids, and let's say this enzyme metabolizes epoxylipids, and these are endogenous and helpful for us to treat inflammatory conditions. So if we block their metabolism, we will stop their conversion to inactive diols, and that could be an alternative inflammatory treatment approach. Since those epoxylipids are generated via cytochrome P450 enzymes, if we could stop the inhibition, the conversion of epoxylipids to diols, then we will be able to keep the anti-inflammatory effects. And let's start to talk about our hit-to-lead journey. And the first target will be FLAP. In the left-hand side, you see the benzoimidazole derivative and its binding to the flap protein. As you can see here, we have a lysine amino acid, which is quaternary charged and it's positive in physiological pH. So if we can put here an acidic functional group that supported the activity results. Later on, we wanted to deal with the plasma protein binding issues that are arising from that functional group. Therefore, we replaced the carboxylate groups with nitriles, and we kept the activity, and we also improved the we dealt with the plasma protein binding issue. Here you see our first hit compound that was found by a virtual screening that's previously done, when we included the nitrile group here at the 5th position of the benzimidazole ring, and also when we included additional functional group on that side by replacing the ibuprofen fingerprint of the compound, then we both kept the activity, the potency, and also improved the physicochemical profile of the compound. So these two papers are mostly about that study that we conducted on benzimidazole groups and the effect of the structural change on flap inhibition based on development of derivatives of BRP7, which was our hit compound. On another study, we see development of isoxazole derivatives, and these series were also found by a virtual screening study, and later on by including a carboxylate group again here, we improved the potency. And this group was not only making an ionic interaction with lysine, but it was also conducting a water bridge with lysine and histidine amino acids residues. So these interactions were supporting the bioactivity of the new compound in the series. And this paper was also published in European Journal of Medicinal Chemistry. In the same study, we also developed 5-lipoxygenase inhibitory effects, which started from 4.4 micromolar and ended with 240 nanomolars. And this was related with 5-lipoxygenase inhibition instead of its activating protein flap. So what we did was to replace the hydroxyl group in the central benzene group and also include an additional carboxylate group here. Then this interaction occurred with asparagine 425, and we were able to improve the activity in this way. As I said, we tuned the structure of the isoxazole derivative, and we achieved both flap and 5-lipoxygenase inhibition. In further studies, we also developed compounds that are active also against MPGS1, prostaglandin E2 synthase. So this could help us to block the pathway both on prostaglandin and leukotriene biosynthesis. So we modified some portion of the amino acids in that region, and by including smaller fragments there, we were able to improve the activity and also keep 5-lipoxygenase inhibitory effect. This work is published in Bioorganic Chemistry. And in the center, we see a benzimidazole-based inhibitor that is more likely to be linear. And these compounds showed MPGS1 and also leukotriene C4 synthase inhibition, which is another enzyme that is located in the leukotriene biosynthesis. So we keep the benzimidazole group from the previous study and replace the other parts of the compound, and we were able to reach MPGS1 inhibitors. This work is published in European Journal of Medicinal Chemistry. In another work, we developed oxadiazole derivatives, and these groups were also active against MPGS1, and we included an amide group here briefly, and this group was acting on a serine residue, which is important for its activity, And by this hydrogen bond, we were able to obtain more potent compounds in the series. This work is published in Bioorganic Chemistry. And here you see, as you can see on the left, we opened the benzimidazole ring system and we reached some sub-nanomolar level compounds that showed also in vivo efficacy. And this study is published in Journal of Medicinal Chemistry. In another work, we improved the bioactivity of MPGS1 inhibitor, and this work is published in Archive der Pharmacie. This compound was previously the core of structure was achieved by a virtual screening study, and by our experience on MPGS1 inhibition, we modified the residues in that, and this in both regions that I am showing here. So with these replacements, we were able to improve the activity of the compound. And here I'm showing a study that we conducted on soluble epoxy hydrolyzers. In this work, we were able to keep important residues, the interaction with the important residues that are catalytically important. So these two tyrosine residues and aspartate here. So these hydrogen bonds were also kept during molecular dynamics simulations. So by keeping this crucial interaction, we were also able to do further hydrophilic contacts here and hydrophobic contacts here with type tryptophan ring and histidine ring system. So in this way, we developed nanomolar level compounds as piperazine urea derivatives, and these compounds were showing low nanomolar effects on the target, low nanomolar inhibitor effects on the target. So all these experiences we gained with the heat to lead journey, we decided to identify new chemical scaffolds for our future studies. In this work, we did a virtual screening study that was applied in a reverse approach. So based on our experience by generating and designing derivatives of FLAP inhibitors, we knew that by docking, we weren't able to directly generate reliable results. But we also knew that by generating some samples of the conformers within binding pocket, we could understand if we were achieving some reliable results, not as top scoring pose, but also by, let's say, sampling 10 different poses per each compound, we were able to generate bioactive conformations. So we decided to generate all the conformations within binding pocket by docking, and we just applied a very simple score filter. And in a million level screening, we just reduced the number and proceeded with 4 parallel pharmacophore model screening studies. And these models were also generated within the binding pocket. So these structure-based pharmacophore models helped us to reduce the number of compounds that are the number of poses that we generated, the number of molecules we generated during screening, filtered during screening, and the remaining compounds were still including a high number structures. So we decided to cluster the compounds based on their diversity and do the selection based on the centroid molecules and their features, mostly their interaction. And then we applied scoring by 3D QSAR models. And this model was also generated within binding cavity. So that helped us to use directly the docking generated pose and to score it within the binding. We also applied random forest-based filtering because we knew that these structures were highly lipophilic. The FLAP inhibitors are very lipophilic structures. So in order to be able to select more reliable but still novel fragments or compounds, let's say, by application of random forest, we selected the top ranking 1000 molecules in there. And later, we visually inspected the resulting poses and did some replacement from the clusters by returning back, eliminating some of the poses and returning back. And in the end, we selected 100 molecules. And by checking the literature data, the patent data that is publicly available, we selected 20 virtual hits based on the results of the MD simulations. We selected 20 compounds and purchased them. And this study showed us 60% success rate. And 4 of those resulting compounds were active at sub-micromolar level. So we were happy with the results published in Journal of Chemical Information Modeling. And if we discuss the biological data obtained in this virtual screening study, as I said in the previous slide, 12 of the compounds were active and 9 of them were also at showing lower efficacy. So the inhibitory effect was higher for 9 of them. And you can see those 9 compounds in green. And as I said, 4 of them was at showing very strong efficacy for very strong inhibitory effect. So from these 4 compounds, we did further biological studies and we realized that we weren't only acting on FLAP inhibition, but we were also inhibiting 5-lipoxygenase. So later on, by doing some literature search, we decided to develop derivatives of these 2 hit compounds. And now I will start to talk about these studies. First, this is the hit compound that we identified. You see the effect of hydrogen bonds here. And by keeping the hydrogen bond and also replacing the position of the polar group from here to there, we flipped the compound inside the binding cavity and kept the inhibitory effect. And in this way, we were able to identify selective inhibitors of 5-lipoxygenase activating protein. And long story short, we replaced the sulfur atom here, but oxygens were decreasing the potential, then decided to keep it. We replaced the position of the polar group and we protected the pi interactions that are occurring here. So in this way, we were able to have good and selective inhibition for FLAP. And this study is published in ACS Omega. In a following study, this was another hit compound that derived from the virtual screening. So you see again the salt bridge and water bridge here. And here we obtained a dual inhibitor by replacing the acidic group with an amide and by also replacing one of the aromatic ring. In this way, we developed dually acting soluble epoxide hydrolase and 5-lipoxygenase activating protein inhibitors. And this quinozoline ring containing structures was able to block the path from 2 different locations. And this study is published in ACS Omega. Long story short, we had 2 virtual screening hit compounds, and with hit to lead optimization studies for these 2 series, we opt for quinozolines, we reached dual inhibition, and for FLAP, we improved the potency and replaced the position of the polars. So let's start to talk about our work that is conducted for MPGS1 inhibition, and that's done by virtual screening. So how we designed our workflow and what were the lessons we have taken from the available data. So on the left side, you are seeing 2D structures of the ligands that are co-crystallized with MPGS1. So you see in the sub-bucket 1, 2, and 3, there are different locations that these compounds are binding. Some are binding only the green sub-bucket, pocket 1, some are binding pocket 2, and some of them are binding pocket 3, the yellow one. So here, 1 is showing that are binding mostly on the packet 1, and 1 and 2 is showing this location, and 1 and 3 is showing this location. So by doing a redocking study, we realized that we weren't able to generate reliable photos. Even by generating multiple conformers, we weren't able to reach reliable results, and especially for 1 and 3 inhibitors, which were showing the most potent effect on the target. So this was happening because there was no membrane structure here, which normally exists in our body, and there were no solvent accessible. There was no solvent effect that was coming from this. So while we were applying docking, even if we keep some of important water molecules, we weren't able to generate reliable results because the lipid was also not here. We generated different conformations for the membrane, their monomers. We kept them for docking studies. We sampled different positions for both the lipid structure and also solvent molecules. But in the end, we haven't managed to improve the binding mode generation for the compounds. So if we cannot dock correctly, appropriately the co-crystallized ligands, we weren't sure how we could obtain reliable results in a visual screening campaign. So we were sure that that wouldn't be possible. So here, with the compounds that are highlighted as green, you see the structures that we focused our studies for the following work. So I will here talk about our virtual screening workflow, how we designed it, and how we made the magic happen here. So firstly, with green highlighted compounds, we generated 3 pharmacophore models, and each of them was generated for this specific series in the co-crystallized ligand data set we had in our. So with the help of these models, we saw that we could sample the right conformer inside the binding cavity. And by generating it inside the binding packet, when we started the conformational sampling after generating the pharmacophore generated binding mode, we saw that we could see better docking results. But it was not enough. But it was satisfying enough to be applied in a virtual screen. But what could help us in a better way? This was maximum common substructure based docking, but it was not easy to apply it in a million scale virtual screening campaign. So what we did, we applied on the workflow A, we applied pharmacophore based, potentially bioactive conformer, redock it inside the binding cavity, and check its reliableness with MCS docking. And if we were happy with the results, then we proceeded with molecular dynamics. And here, by the path A, we did a million scale virtual screening study. And by filtering millions of compounds, first with Payne's and Rios, and then modified Lipinski and Weber filters, we reached low micromolar level compounds, hit compounds, and in total we tested 34 chemical compounds, chemically distinct structure containing compounds, and this work is published in Journal of Molecular Graphics and Modeling. And on the path B, which we haven't published it yet, we generated the binding poses by AI-driven solutions. By the help of diffusion modeling and some other approaches behind, we generated the right poses without needing pharmacophore models and also without needing maximum common substructure based. And docking, we were able to dock properly the co-crystallized ligands and also some potent MPGS1 inhibitors that is included in our internally curated data set, which contains 8,000 data points for MPGS1. And with the help of this data, we also stressed the target-specific scoring performance of those compounds. And by applying molecular dynamics, we ended the study. And as I said, the AI part of this work hasn't been published yet. But what was the difference in between both studies? Here on the left, you see the cross-talking study that we did with Glide, and on the right, you see the cross-talking study that we did with DiffTalk. And inside this heat map, you are seeing the RMSD values of both works. On the left, you see the RMSD values are very high, all are above 7. The averages of each solution was above 7 angstrom. And this is something that we cannot rely or study in a pure docking approach. But with the help of AI, we were able to obtain much reliable RMSD values, which were mostly below 2 angstrom. But we were still seeing some issues with some of the compounds at very small level. But we said that will be acceptable for us. But would it be enough to generate a binding pose within the binding cavity? And if you don't see a scoring performance that is able to distinguish the most active ones from the inactive ones, or intermediate level actives, middle range actives, if you cannot separate them with a score, then it will be again difficult to understand which compounds are more reliable to be tested in our biological laboratory. So we applied different approaches, and as you can see in the green rock curve, we were able to distinguish the most active compounds from the decoy sets and also inactive ones. And the magic happened here by the help of Nina. So Nina directly wasn't working properly. There was a memorization issue that was occurring with Nina. But when we did the docking with DiffTalk, but only minimized the generated pose with Nina, then we were able to successfully score the compounds that were already generated within the binding cavity. So with the help of Nina and minimized RMSD function of the docking solution, we were able to reach good results. So here I will talk about soluble epoxy hydrolase inhibition. These compounds are, as you might remember, showing key interactions with these two tyrosine and one aspartate residue here. So soluble epoxy hydrolase has two functions. One is the epoxy hydrolases, and the second one is the phosphatase effect. So we were targeting the hydrolase effect. So this catalytic triad opens the epoxy ring, and with the help of tyrosines, the rings open, and the epoxy hydrolases effect is achieved by this enzyme. So when we place a compound that competes with those epoxides and inhibits the enzyme, then we will be able to protect the endogenous epoxy lipids that are in our body. And then in this way, we will be able to have anti-inflammatory effect by inhibiting the hydrolase effect of the enzyme. And for this work, we screened million and billion level compound libraries. And to generate a virtual screening workflow, we created data points from different data sources, and also we did manual curation, tested the hypothesis, which I will not be able to go into the details of the work, but by identifying the right virtual screening workflow, we applied both, again, physics-based and AI-based virtual screening studies here after doing physicochemical filtration. Then we did the final level of compound selection with ADME and toxicity prediction. And by utilizing molecular dynamics simulations, we selected the compounds and started doing in vitro experiments. And again, heat maps were very helpful for us to select the right docking solution, and also the target scoring performances were very important. We screened million level of compounds and billion and also trillion level compound libraries, and we reached very good results. So how did we generate the workflow for the study? So here on the left side, you see the cross-talking results that are generated with 112 crystals of soluble epoxide hydrolytes. As you can see here, the green values show good results and the red ones show bad results, and mid-range is shown as yellow. So with Vina, we were able to generate very high quality poses, and we were also able to interact with the important residues inside the cavity. And also with FlexX, we were able to achieve very good results, as you can see in the green data points, with green RMS. But there was only one issue, and it was related with either docking related or RMST calculation related issues. We weren't able to fill all the data points here, and these are shown as white. But very briefly, if Flex was generating a pose, then we saw that that will be reliable. And if we apply this approach, if we are able to apply this docking solution in a billion and trillion level compound library with chemical space docking, then we can achieve very good binding modes for the compound. When we applied the same study with Glide with different scoring functions, we were able to reach middle range and also good level of good results for the binding events for the cross-docking study. But then we wanted to do this. We also wanted to use this solution in million scale screening, but we haven't prioritized it during compound selection. And here you see the results of AI-related tools. And on the left-hand side, you see the heat map of DiffDock, which is looking very good, but this may also be related with the training data, since most of the crystals were also included in the training stage. We wanted to be very careful about the data handling. And the same was also for Nina. The results were good, but we were not sure if these good results were coming from memorization. But we stressed it, and we saw that most of the good results were not included in the training stage, and some of them were chemically very distinct. So we felt that we could use both solutions in a virtual screening campaign. But how would we do that? As we did for MPGS1, we again preferred to use DiffDock for pose generation and Nina for scoring, which I will start to talk about it. And then we also stressed the newly emerging co-folding option, AlphaFold 3, Rosetta Fold Olatum, and Boltz 2. They were able to generate very good poses, but they were taking a lot of computational resources. And also, since this is a co-folding solution, these are co-folding solutions, we realized that, and we were also expecting that in the end, we saw that it was not so easy to filter the compound, because first of all, these tools, AlphaFold 3 and Rosetta Fold Olatum, wasn't including a scoring function, as in the case of DiffDock. But since the solution was computationally intense, computationally demanding, and also since the results were not so good compared to other solutions, we wanted to see the induced fit effect as we were evaluating in molecular dynamics stage. So we decided that we could use these co-folding solutions not in the filtering stage, but right in the last part of the study, we could use these tools in parallel to molecular dynamics, and we could understand the molecule-induced binding events inside the cavity. Here you see the scoring performances of all of these solutions that we discussed here. So on the left, you see the ROC curves of Vina, and in the middle, you see the ROC curves of HTVS, and on the right, you see the rock curves of glides. So here, the dark greens are the most potent ones, and the red ones are the most inactive ones in our data. And the others are mid-range. So all these tools could easily differentiate the actives from the inactive, including the decoy. But the best one from these three options were Vina and Glides, as you can see the areas on. When it comes to FlexX, the results were very close to be random. Therefore, we decided that filtering with directed docking with FlexX would not be an option. We wanted to be able to dock billion or trillion scale of compounds in our virtual screen, and we already found a solution, and I will discuss about it in the following slides. But when it comes to DiffDock and Nina-based scoring, the area under curve was 0.86, and it was very good, and it could distinguish again the actives from the inactives, and the results were looking very logical. So we also decided to use this combination for the bridge. And as I said, AlphaFold and RosettaFold Olatom was not including a scoring function that could be used to compare between different like. We applied Nina-based scoring for these tools, but the results were not very good when it came to both, either by using Nina-based scoring or also their internal scoring function. Again, the dark green lines were performing much better, and the red and orange ones were, as expected, were showing low areas under curve. So for the dark greens, the results were almost 0.9, the areas under curve, for both Nina-based scoring and also both tools internal scoring function. So then we wanted to do the final evaluation with this tool. And I'm returning back to Flex. As you might remember, that we weren't able to reach reliable scoring scores with Flexes, but we wanted to be able to screen billions and trillions of compounds by docking. Therefore, we decided to use structure-based pharmacophore models and do a docking in parallel. And we applied when we developed these different models, we were able to reach very high area under curve value, and we decided that we could use these values in a virtual screening campaign at trillions. And the pharmacophore models also helped us to screen the library even faster than directly using docking. So we could both achieve good results in terms of binding mode generation, and we could also score much more better. And here you see a comparative study between the most actives and inactives, and also mid-range compounds, and by using pharmacophore-based solutions, either by phase or FlexX, we could achieve very good results. But the only issue was coming with this, it was related with the scoring performance. But we should note that the decoys, most of them were not able to dock inside. So even though we cannot generate a right score comparison for these compounds, we will still filter out a high number of compounds that are expected to be enough. Therefore, we were pretty sure that we could use pharmacophore and docking approach together with FlexX to be utilized in chemical space docking campaign. And in this way, we could screen billion and even trillion level chemical libraries, and we could simply filter those huge databases with chemical space docking and reach 1,000 and million scale results. We could also keep even less compounds just by playing the parameters of the software. But we were already sure that we could apply reliable virtual screening campaigns at million level. So the filtering of trillions to million was already more than enough for us. So we wanted to stay here and proceed our work. So this was Emine Yekta Yılmaz's work, my Master of Science student, and she did a great job there, and she was also in Winter 2024 Challenge of Biosol IT, and by their software donation and their support, Yekta did a tremendous effort, put a tremendous effort, and did this work with the help of Didar, and also for the biological work, Simla is helping a lot. This study is still ongoing, but we are pretty sure that we will be able to identify very potent and chemically diverse and very valuable results at the end of this spring campaign. So I believe I am right on time, and I can start thanking my university, to our department members and our research group members, Banolu Research Group. And I would like to start by thanking Professor Dr. Erdan Banoğlu and Professor Dr. Burcu Çalışkan. I also would like to thank Professor Andrea Caruti from University of Perugia for his help, and to Professor Dr. Oliver Wers from Friedrich Schiller University of Vienna from Germany, and his team is doing a great work for us, and we are very happy to collaborate with them. We would like to thank Avias Pharma. We would like to thank Biosol IT for their free license access, and also the chance to speak here. We would like to thank Radius Computer especially. Their hardware donation helped us to improve our CPU-based computational power. They donated more than 300 CPU cores, and this was very helpful for our huge screening campaigns. We also would like to thank the chipset project support received, and we would like to thank the resources that we were able to obtain, the computational resources that we were able to obtain from both Turkish Science East infrastructure, the Turkish supercomputer, and also we would like to special thank the Barcelona Supercomputing Center. And also we would like to thank all the grant support that we achieved for this work that I presented. And before concluding, I would like to quote the founder of the Republic of Turkey, Mustafa Kemal Atatürk, who said, "If my words go against the science, choose the science." With that, I would like to thank you all for listening, and thank you very much.